Hi, my name is Sheila Rao, and I am welcoming you to the first module in the LEND program. I am going to run um, through an overview of a lot of different genetic disorders. So some of the learning objectives that I'm hoping you walk away from this presentation include identifying and using common genetic terms. So we're going to go through a fair amount of terminology. We're going to understand a little bit more of the differences between genetic screening and actually definitive diagnosis, have some more of an appreciation of the impact of the environment on the development and the variations of genetic disorders. So I am really working from the public health framework known as the eco-biodevelopmental model. And lastly, I will be introducing some resources that hopefully you can explore with families for more genetic information as the need arises. So when it comes to thinking about pediatric genetics, 2 to 3% chance for any couple to conceive a child with what is considered a major birth defect or a major genetic difference, and 3 to 5% for a minor genetic difference. Over 33% of all pediatric hospital admissions are due to genetic causes. What is genetic? So genetics basically is a term that refers to the study of genes and their role in inheritance, the way certain traits are passed down from one generation to another, really looking at more the way that one gene may cause a particular or be linked to a particular condition. Inherited means that a parent has a genetic trait or difference that is being passed on to the offspring, but you can have genetic conditions that are not inherited where a person has a spontaneous genetic difference that their parents don't have. And congenital just means that a condition has existed since birth. When we're looking at more condition definitions, genomics, on the other hand, is a much more comprehensive term. So we're looking at the study of all of a person's genetic material, the interactions of that genetic material with itself, the interactions of the genetic material with this interesting concept known as the environment, and the subsequent phenotypic changes or variations that will change a person's life course. Epigenetics really looks at like these processes that can be occurring above the level of just looking at the genes and their roles in inheritance. So even though Conrad Waddington came up with this concept a long, long time ago, these processes really are much harder to get an understanding of, and sometimes we don't fully have a complete appreciation of these, the roles that these processes play in the way that disease marks itself over a person's life course. So while genetics may use information from one or two genes to explain a disease, genomics really strives to show that all of the genetic information that can possibly influence biology, predisposing an individual to a disease, should be included in looking at the aftermath of a genetic difference. So how do we inherit? When you think about the basics, we start off with 4 nucleotides that form this double helix. These basic building blocks, these nucleotides are our DNA. And we have millions of these nucleotides strung together in very distinct packaging known as chromosomes. So if you can think about it this way, if we've got like a million base pairs making up 22 different books with one set of books known as the sex chromosomes, and each individual has a characteristic set of groups of building blocks known as genes within each book of our DNA. So overall, we have now sequenced the entire set of millions of letters that make up the human genome, and we've come to determine that about 21 to 25,000 genes are arranged in this particular sequence across human beings. And overall, this pattern has stayed distinct across our species. So when we think about looking at the sequence of our DNA and comparing it to other organisms, we actually have a fair amount of similarity across many different organisms. So zebrafish share 70% of their DNA with us. They are often used as great models for early development because their eggs develop outside of their mothers. Mice share about 90% of their DNA with us, and they too are studied very extensively in genetics because one mouse year is equivalent to about 30 human years. When we look at dogs, they only share about 84% of their DNA with us, though our relationships with dogs seem to be very, very tightly evolutionarily matched. We are often using dogs for studying diseases like cataracts and retinal diseases because those diseases affect both dogs and human beings. And then lastly, with chimpanzees and bonobos, we share about 98% of our DNA. So if you think of it this way, well, what could account for these differences? Some genes do get turned on and off at different points after conception. So although, if you think about that 98.8% similarity in the sequence, it's probably not so much about looking at the order of the nucleotides, but rather looking at the parts of the DNA that do not necessarily code for genes. So for instance, when you think about the course of expressing a gene, there are several processes, transcription and translation, that occur to change a gene to a protein. And there are environmental cues that signal whether or not transcription, translation are going to take place. Also, what we've come to realize is that these cues may also trigger what is known as alternative splicing. So you may have the same set sequence of nucleotides on a part of your chromosome as a dog or as a chimpanzee. However, we may have more sophisticated mechanisms for transcribing and translating that sequence into multiple different proteins. Or we have more parts of our DNA that control the splicing of these sequences into proteins. So even though we have about 25,000 genes, really overall that only makes up about 2% of the human genome. And so most of the human genome are non-coding nucleotides. So the average human gene will have about 1 to 3 nucleotide-based differences from one person to the next. We really don't have more genes in previous animals, but we probably have more sophisticated control of them. So how do these genes change over a lifetime? This is where we really look more into the role of epigenetics. And these are some videos that you can take a look at. Overall, we have identified 3 different ways that we can change the ways that these sequences are transcribed and translated. So one system is known as DNA methylation. So somehow, again, the environment will add methyl groups in certain places on the genome and therefore cause different transcription and translation of the DNA. Another system is known as histone modification. So histones are packaging systems for our DNA. You can think of it as the ways that we tie up our DNA and like wrap them together into like really, really tiny chromatin. And whether or not a gene gets exposed to messenger RNA to get transcribed or translated can depend on the way that it is wrapped or unwrapped. And these histones will change their the way that they are orienting towards the mRNA depending on environmental cues. And then lastly, when we look at the non-coding parts of the human genome, they actually play a huge role in silencing certain genes. And so looking at the environment of the cell, both inside and the outside, is becoming more and more important to the understanding of genetics and genomics. We get signals that indicate alterations in homeostasis. Your cell triggers a cascade of genetic consequences from these changes. So this is another video you can take a look at. Another concept that you might see that can also alter the way that a gene is expressed is imprinting. So on two homologous chromosomes, sometimes one copy will get silenced from one parent, and the only other copy will be expressed. And there are about 30 genes that function this way. And what is very interesting is that there is often a very specific copy that is indicated for this gene expression. So it might be the maternal copy for certain genes and the paternal copy for other genes. When it comes to thinking about these epigenetic changes, we still have a lot to learn. When we look at cancer genetics, for instance, we've come to just realize that there is a huge role that the environment can play in turning off and turning on genes that relate to cancer. So we've seen that there are carcinogens or cancer-causing triggers in our environment, such as ultraviolet rays or irritants like cigarette smoke. Those genes that are related to tumor suppression might be turned on by getting those environmental cues accumulated over time. And then we have changes at the base pair level that may cause different coding to occur. When we look at the types of genetic disorders, classically, we have a lot of capacity to look at number differences. So aneuploidy just means a difference in number, whether it's a trisomy or a monosomy. Translocations, deletions, and mosaicism are a little bit more sophisticated to explore in terms of figuring out whether or not a person has these differences. So translocations may include switches of pieces of one chromosome with the other. Deletions are when parts of the DNA are removed, duplications when they are duplicated. Mosaicism indicates when cells in an organism have different genetic makeup. So one cell might have a genetic difference and other cells don't necessarily. And this will impact whether or not the severity of a particular disease is being expressed throughout the organism in the same way or not. We also have a fair understanding of single gene disorders, and I'll show you some of the patterns of inheritance that we've come to understand fairly well for these sorts of disorders. And so this is where perhaps a base pair difference has occurred spontaneously or not spontaneously, but needless to say, over time, those patterns of inheritance, once one individual in one generation has inherited this difference gets passed on in specific patterns. Lastly, there are genetic disorders that might be linked to mitochondrial DNA. So when we are creating sexual cells, the gametes, the eggs and the sperm, they are receiving half of the DNA from the parent. And mitochondrial DNA is only inherited from the mother. And as a consequence, you are more likely to retain and inherit any differences that have occurred at the mitochondrial DNA level. When we look at these different types of genetic tests, we use different tests for different things. So chromosomal analysis or karyotype is sort of our airplane view of how many chromosomes do you have? Do you have the typical number or not? Chromosomal microarray basically will tell us, are the sizes of your chromosomes more or less consistent with what is expected? So we can get a ballpark idea if there's some deletions in some places, but oftentimes we won't get the details that we would obtain from other tests to know exactly where those genetic differences, if they exist, are. There are more explicit tests when you are looking for a protein difference that has occurred because of a change in the DNA. So when we're looking at plasma amino acids or urine organic acids, we might be looking for the accumulation of certain proteins that are occurring in someone who has a gene that codes for something differently and therefore changes chemical processes in that person. Lastly, we have the capacity now to look at the actual sequence of the DNA on the human genome and look for single base pair differences depending on the clinical features of a person's condition. So there are panels, for instance, for epilepsy, panels for specific types of conditions that will pinpoint looking at the base pairs in maybe 5 or 6 different chromosomes. And to identify if an individual actually has a base pair difference that is known to be causing a particular disease or condition. So when we think about some common examples of chromosomal aneuploidies, Down syndrome is very well established as a known chromosomal abnormality. Typically, most individuals with Down syndrome do not inherit this condition from a parent. It is typically happening at the point of cell division when the egg is being created, that rather than having only half the number of the chromosomes, for some reason, there's an additional chromosome 21 left in the egg. Typically, there's a low recurrence risk for trisomy 21. However, we have seen that overall, over time, mothers with advancing age, which is defined as 35 and beyond by the American College of Obstetrics and Gynecology, will have increasing risk. And there are some very specific clinical features that are now known to be identified with trisomy 21 because chromosome 21 is involved with heart development and intestinal development and muscle tone. So there are some specific red flags that we will take into consideration when we know an individual has trisomy 21. So if you look at this is what is known as a karyotype. So again, this is your airplane view. How many chromosomes do you have? Are they the typical or atypical number? And here you see the 3 chromosomes in the chromosome 21 section. And this individual has an X and a Y. So this is a male who has trisomy 21. Trisomies are very different depending on which chromosome is affected. Trisomy 13, by contrast, is associated with a very severe brain abnormality known as holoprosencephaly. And 90% of individuals die within the first months, if not the first year of life. There is a low recurrence risk for trisomy 13. However, for the most part, most common chromosomal abnormalities will end up as miscarriages, such as trisomies, triploidies, or monosomies. The incidence of chromosome abnormalities among spontaneous miscarriages or abortions is about 33%. So the most frequent chromosome abnormality that is actually compatible with life is trisomy 21. And about a little bit more than 1 in 1,000 individuals are born with trisomy 21. And of course, this prevalence statistic may be changing over time with the changes in population epidemiology, but needless to say, overall, chromosome abnormalities in terms of aneuploidies are often individuals with these sorts of differences have a very different life course depending on the chromosome involved. Other things that I want you to keep in mind as we're exploring different sorts of genetic conditions is that overall, again, our sequence of DNA only codes for about 25,000 genes, but less than 1% of these genes are slightly different among people. So you very likely have a very similar chromosome sequence to the person next to you, even if they're not related to you. Modern humans have only been around for about 150,000 years, so we don't really have any genetic differences that define race. There's no such thing as a human subspecies. And when we're really thinking about genetic origins of health and disease at the population level, you have to appreciate the impact of migration and also what is known as geographic and genetic isolation. So whether or not populations continue to marry and have offspring over time, disease frequencies can change depending on the isolation of different populations. So when we think about common experiences of human history, such as temperature, latitude differences, we'll see that that can oftentimes trigger environmental cues that perhaps change coding for melanin content of skin or vitamin D requirements. What we see are that migration patterns do lead to visible differences, but not at the genetic level. We're still 99.9% the same. What I do want you to get an idea of, though, is that there are changes that can occur in the frequency of certain disease carriage in certain populations depending on these environmental experiences that they have. So say you have a parent population perhaps on a different continent and there's a migration to an island somewhere, or perhaps there's a massive epidemic of a really lethal disease such as influenza or the plague. There, for whatever reason, if there's a huge reduction in the population, the frequency of certain genes might be very different in the subsequent generations who survive whatever that bottleneck experience was than the original parent population. And so when you think about, for instance, the selective force of malaria, on the left are all the regions in the world where malaria is prevalent. So the darker the green, the more malaria there is. And of course, this tends to be around the equator because of the way that mosquito populations are continuing to evolve. As far as the carriage of sickle cell carrier, we see that it also follows a very similar pattern and tends to have a fairly high prevalence around the equator. And the reason why this particular carrier has continued to remain high in frequency in populations that have malaria endemic is because there's a selective benefit for carriers of sickle cell when it comes to surviving malaria. So carriers of sickle cell allele do a little bit better with malaria than people who don't have that particular genetic allele. Same with cholera. So we've seen that when you're looking at particular populations and trying to target your identification of a particular disease, you're going to choose your genetic screening depending on where in the world you're seeing higher frequencies of certain alleles. So the condition known as cystic fibrosis is worldwide prevalent, but there are certain chromosomes and certain alleles and certain base pairs that tend to be frequently identified in European populations versus populations of the Americas versus populations in other parts of the world. So we try to target our testing depending on the population of interest. We recommend referral to geneticists whenever any newborn has a genetic screen that is being identified as abnormal. So we might not necessarily be capturing a disease with that abnormal newborn screening. However, we are identifying that there might be perhaps some sort of airplane level genetic difference, but we don't have the specifics of that. When it comes to seeing the development of a newborn, any concerns of muscle tone differences, hypotonia, known birth defects identified at birth, such as cleft lip, cleft palate, congenital heart defects. These are all reasons to connect a family to further genetic testing. In California, we have very comprehensive newborn screening with over 80 conditions. We've even included rare conditions like mucopolysaccharidosis. 99% of the babies that are born in California are screened, and we have specific centers that will clear out these tests depending on the birth hospital where these babies are born. And we'll be talking a little bit more about newborn screening. These are some of the other conditions associated with newborn screening. And when we think about patterns of inheritance, for the most part, we're capturing a lot of classic Mendelian single gene disorders, but we'll eventually be moving towards non-Mendelian inheritance patterns that really involve more of the epigenetic processes that I've mentioned before. So I just want you to be familiar with some of these patterns of inheritance that come along, particularly with single gene disorders. So a lot of times you might see, for instance, with autosomal recessive inheritance, that there is a predisposition towards autosomal recessive inheritance if there's a known background of consanguinity in a family. Another thing that you might see is that perhaps in the generation of offspring, there's about a 25% chance of a generation member being affected, with about 75% not affected. Carriers don't typically have any clinical features of the condition, whereas in autosomal dominant, you have a 50-50 split with the subsequent generation, because any individual with carriage of the gene will have clinical features of the condition. So this is a very different pattern of inheritance. Another thing that you'll see is that there might be what is known as incomplete penetrance. And so individual by individual, they may have different features of the condition, even though they theoretically only are carriers. With X-linked conditions, though, what we tend to see much more is that there's mother to son transmission, with daughters having a 50-50 chance of being carriers. And you don't see father to son transmission of these sorts of conditions. There's no male to male transition. When it comes to looking at the ways that we can treat genetic disorders, we are really thinking more and more carefully about the type of environmental cues that may be triggering coding of gene products. So diet and medication may really change the outcomes for certain individuals. Fatty acid oxidation disorders, you might have to just avoid situations of fasting, which can trigger these sorts of gene products to accumulate and cause negative outcomes. Sometimes we modify genes in the ways of giving other people's genes to an individual who might be missing a particular X-linked condition. So, for instance, in severe combined immunodeficiency, stem cells transplants are an option if we're able to capture and identify that an individual has this condition early enough in their life course. And then last but not least, sometimes we're not able to treat a particular condition, but we might be able to mitigate secondary effects. So again, thinking about the red flags that I brought up earlier, getting that early referral to geneticists and genetics multidisciplinary centers might be really pivotal in changing the outcome of a person's life course. So keep this in mind whenever you're seeing an individual who is manifesting developmental delay. Failure to thrive is poor clinical descriptive term, but really indicates that a person is not growing as we would expect in their, or they might be losing milestones that they previously had acquired and established well. Weakness of muscle, vision and hearing loss, sensory losses tend to have a genetic basis. And so we will always look and send referrals to geneticists the sooner than for other conditions. And now we're casting a wider net for genetic differences. Our developmental behavioral pediatricians do recommend getting a genetics referral for individuals who have autism spectrum disorder, growth issues, learning disabilities, epilepsy, ataxia. I already mentioned that there are panels where they'll look at specific groups of chromosomes together to see if there are genetic differences that might be causing the seizures that we're seeing. And then, of course, with fetal alcohol spectrum, this is a very unique spectrum of clinical features that often are occurring because of environmental cues in the in utero environment. So our genetics professionals include many different disciplines. We have genetic counselors, geneticists, and dietitians, as well as social workers. And there are special referrals to regional centers indicated for specific genetic conditions. And there's a huge host of genetic conditions that qualify individuals for CCS, and possibly additional therapies, resources, clinical trials that they would not normally be able to access if they weren't identified to have this genetic condition. So this is why early referral is really important, because the process of diagnosing genetic disorders can be an odyssey for some families. The genetics clinics include multidisciplinary and specialty clinics, such as the ones listed here, and a lot of them are getting funding at the national level from the Genetic Handicapped Persons Program. So those are additional resources that may help to target therapies and potential treatment approaches for individuals with a specific condition. So it is very important for families to be going to centers of excellence for a particular condition. Beyond pediatric genetics, we are now even looking at preconception carrier testing, preconception genetic diagnosis, prenatal non-invasive prenatal testing. So, for instance, taking blood tests from mom during pregnancy to identify if there are genetic differences in fetal DNA. And that also includes CVS sampling, which is much more invasive, and amniocentesis. With our advances in genetic testing, we have also much more capacity to identify conditions that might affect an individual, not immediately in childhood necessarily, but maybe even later on, or we can pinpoint things that we have to look for because we've learned that a person has a particular condition like Marfan syndrome or a predisposition to heart arrhythmias. So there's all sorts of different genetic tests, and now we've also seen more and more consumers using recreational genetic testing. And of course, we have to explore more and more the ethical issues involved with these sorts of tests. So you might be looking at late onset disorders that will never affect individuals in their childhood, and whether or not we are opening a Pandora's box is truly up for debate. Testing adopted children, because we don't have any genetic history from family members, just to identify ballpark idea, again, airplane level, like, oh, does this person have a predisposition towards certain genetic disorders or not? And whether or not late onset disorders are precipitating early, pregnancy terminations is also up for debate. And then, of course, now that we are getting more and more sequencing of populations of people, whether or not this data sharing is going to impact the way that we interpret the tests that are available to us. So we have numerous resources that we encourage you to take a look at. I've included these different websites and the National Society of Genetic Counselors website as well to explore some of these topics. And again, thinking of individuals, these two kids are siblings, and the little boy has Russell Silver syndrome. And so they are probably most genetically similar to each other than they will be to anybody else on earth. Siblings have the same parents, and so they oftentimes will have the most homology of sequencing of their genetic base pair, DNA base pairs. However, the burden of the genetic conditions that they are predisposed to are going to be very different over the life course of each one of these kids. And so we really have a lot of work to do to really appreciate the ways that our carrying a condition over the course of the lifetime is going to change. Thank you very much.